if you don't mind, we will record this talk, and this video will be posted on the YouTube.
Right.
Okay, thank you. And before you start, I will give an introduction. Okay.
Okay, sure. And then I'll share the screen, right?
Yeah.
Okay.
You can share your screen right now.
Okay. Let me do that.
Let me stop sharing my screen. Okay.
Okay, you can see my screen.
No. Now we can see your slide.
Yeah. Just let me go to the next slide to see if the videos play, okay?
Okay.
You can see it play, right?
Yeah.
Okay, I'm good.
Okay. So let's get started. Hello everyone, welcome to our Nanomotor mini talk. And it's my great honor to introduce our today's speaker, Professor Ayusman Sen, to give talk on our mini talk. I believe we are all familiar with Professor Sen for his great contribution to the research field of micro nanomotor. Professor Sen is a development professor and distinguished professor of chemistry and professor of chemical engineering at Penn State University. His research includes active autonomous systems, synthetic nano micromotors, micropumps, and nanotechnology and systems chemistry. The Sen Lab was a pioneer in the early days of the development of chemical nanomotors. In the more recent years, his lab has been leading in the research of enzymes as active matter. During his research career, Professor Sen has received numerous awards and recognitions, such as the Langmuir Lecture Award from American Chemical Society and the Humboldt Prize. He is also an elected fellow of Royal Society of Chemistry, as well as the American Association for the Advancement of Science. Professor Sen has published more than 700 papers with H index of 94 and more than 30,000 citations. So without further ado, let's welcome Professor Ayusman Sen to share his view about Nanomotor Roadmap. So please go ahead, Professor.
Thank you. Thank you for the kind introduction. And thank you, Wei, for inviting me to give this talk. So this is, we insisted that this would not be a, there is some noise in the background. Maybe the others can mute for the moment.
Yeah. I'm mute.
So we insisted that I don't give a straight research talk, but talk about my perspective going into the future. As Yogi Berra, and you probably don't know who he is, he was an American baseball manager, and he used to make very odd sayings. And so this is one of the sayings. It's tough to make predictions, especially about the future. And of course, the future is where you make predictions. So in any case, this is going to be a personal view, and I'm sure I'll leave out large areas where people are very active and doing wonderful work. But I haven't thought about all the aspects of nanomotors, so you will excuse me if I leave out certain things that might be your favorite. So the grand challenge in this area is to design active systems. Let me do something here. Yeah, to design active systems that show emergent properties through interactions. So things that are driven out of equilibrium by input of energy, and they interact with each other, they interact with the surrounding, and they act independently. So this requires two components. It requires something that is active, that can move around, that can sense information, that can harvest energy. And then for it to be programmed, you need a source of information. And today I'll only be talking about chemical motors, because that's what I know well. There are wonderful work in magnetic motors and in acoustic motors and other forms of propulsion, but I'll just focus on chemical motors. So in our case, for chemical motors at any rate, the information comes in the form of gradients. It can be chemical gradients, it can be light gradients if it's a photocatalytic reactions and so on. So we have a system that is able to be active and pick up on information and harvest energy and a source of information. And these two are both essential for functional nanomotors. Now, one thing I should stress is that it is always possible for things to move and act by an externally imposed field, like an electric field if your particles are charged, magnetic fields if your particles are magnetic, but these are defined, the motion is defined by the field. So it's a lockstep motion, and so in my definition, these are not motors. The ones that I'm talking about have self-generated fields. They harvest energy locally, they make decisions locally, and this requires, for the most part, catalysis, either straight catalysis or photocatalysis for this to happen. And so that's what we call motors, things that self-generate their own motion. Now, just a simple idea of a roadmap, the field of materials started with passive nanostructures, coatings, polymers, nanoparticles, etc. Then it moved to active nanostructures, so things like actuators, amplifiers, adaptive structures, and so on. And what I see the field is going towards is systems of active nanostructures. So it's not just individual nanostructures, but systems of nanostructures. And the overarching goal in the field is to accomplish tasks that you cannot do any other way. So it's no good if the nanomotors are a slight improvement over other methods or tools that are available now. The nanomotors need to accomplish something that is just impossible to do at the moment with any other means. And so what we need is a killer application. And while we're on the subject, let me try to sell you the idea of systems materials. So you've heard of systems biology and you've heard of systems chemistry. And I want to talk about systems materials, which is the study of interaction and feedback between individual units of an active system, which gives rise to the function and performance of the entire system. And so some of the very important questions that need to be addressed moving forward in this idea of systems materials is, do you need these systems to be based on modular structures? So discrete autonomous units that come together. And if they come together, how do they come together? Do these units decide on their own to assemble themselves based on their interactions? Or do you need an external field and program to cause their assembly and function? And I think the most important is the point number 4, which is that you need a source of information. And where in the system is this information stored and how is it communicated? So in systems biology, for example, the information is stored in the DNA. Here, the question is, where is the information stored? And how can these nanomotors access the information and act on the information? So I want you to think about systems materials and these interactions between different components of these systems materials. Now, so what I thought is that what I would do is list some of the possible interesting applications that are unique to autonomous systems, nanomotors, etc. And I list them here. And what I'll do in the rest of the talk is to talk about these one by one. So the first one is motion-based targeting. This is cargo delivery, for example. This is to direct particles to specific targets while the sample of fluid space. And because they can move around, they can sample a wide area, and so they don't get trapped in local minima. They can find their targets. This is a really active area, and I don't really have much to talk about because there's so much work going on here. I just show one example where you see these active particles carrying cargo, for example, this middle right-hand side. And this brings me to an important point, which is that in many cases, the cargo is too large to be carried, transported by a single nanomotor. And so there you need assemblies of nanomotors that can communicate with each other and carry the cargo as a whole. And this is a direct analogy, for example, in animal world, for example, ants, often they carry large insects that a single ant cannot carry, and they act cooperatively to carry them. And so one needs to start thinking about using assemblies of nanomotors to carry large cargo. This is particularly important in biomedical fields because often to get a proper dosage of drugs, you need significant amount of drug, and that cannot be accomplished by single nanomotors. I want to talk a little bit about enzymes. We work in this area, but more to the point, it illustrates some of the other applications that I want to talk about. So by now, I think all of you are aware of the fact that enzymes, when they catalyze reactions, they generate force. And these are just simple enzymes. These are not motor proteins. And the force translates into mechanical motion. And so for enzymes, for example, this is manifested in enhanced diffusion. And as you can see for catalase, for example, as you increase its substrate concentration, which is hydrogen peroxide in this case, the diffusion increases with the reaction rate. Now, the mechanism of this is unclear, and in fact, the extent of the diffusion has been controversial in the literature. So I won't dwell too much on it, except to say that it is clear that there is some enhancement in diffusion. It is also clear from work of many people that you can attach enzymes to larger microparticles. By catalyzing reactions, you can cause these microparticles to move. So clearly a force is being generated when enzymes catalyze reactions. And you can do a back of the envelope calculation. It's about 10 to 15 piconewtons per turnover. The other interesting thing about enzymes is that they show chemotaxis, which used to be something that only living matter showed, but we were the first to show that, in fact, catalysts in general tend to move up the substrate gradient. So here is just one example where you have a 3-inlet microchannel and you have urease, uniform concentration of urease in all 3 channels. And if you pass urea, which is the substrate, then the urease from the flanking channel go into the middle channel, so they focus. So you go from an equilibrium distribution of enzymes to a non-equilibrium distribution of enzymes. And you can do the same thing with enzyme-attached particles. You can make them chemotax by providing a gradient of the substrate of the enzyme. So that's an example of information in the form of gradient that is directing the motion to a specific area or direction. Now, and you can use it for various applications. This is just one example where you can use this idea of chemotaxis to separate enzymes based on activity. So you can pass a mixture of active and inactive enzymes through one channel, the substrate through for one specific example through the other channel, and only that enzyme which becomes active because of the substrate turnover will migrate towards the substrate and will come out. And we show this with active and inactive catalase, for example. And so this allows separation of proteins that are otherwise identical in charge and size. So the second application I wanted to talk about is this idea of catalysis in catalytic cascades, and that chemotaxis promotes substrate channeling. So this is one way. If you have a multi-step catalytic transformation and you want to speed it up, then chemotaxis is a way of speeding it up. And this happens in your body. For example, you have these enzyme cascades. For example, the glycolysis cascade that converts glucose that you eat into ATP at the end. And there are some 20-odd enzymes that are part of the cascade. So the first enzyme reacts with glucose, makes a product that is the substrate for the second enzyme. The second enzyme makes a product that's the substrate for the third, and so on. And what the biologists have found is that if you have these, if you provide the substrate for the first enzyme, all the enzymes in the substrate, all the enzymes in the cascade come together. And these are called metabolomes. And here's an example. This is not from glycolysis cascade, but purine synthesis cascade. And you can see actually that these are really large clusters of enzymes that are part of the cascade. And the nature does it because it's much more efficient to just keep handing over to the next enzyme and the next enzyme and the next enzymes to speed up the reaction. You also have less wastage because the intermediates don't get lost in the bulk. And if the intermediate is unstable, it doesn't have a chance to decompose and so on. But the important question was, how is this happening? How are the enzymes coming together? And our hypothesis was it's just chemotaxis. So the first enzyme makes a product, which is a substrate for the second. So the second will chemotax towards the first. And then the second makes a product that's a substrate for the third. So the third will chemotax to the second. And so they all come together. And we looked at this. We looked at the first four enzymes of the glycolysis cascade. Hexokinase, phosphoglucose isomerase, fructokinase, and then finally aldolase. And you can, again, set up a microchannel where you have the first two enzymes in one flanking channel, the second two in the third and fourth enzyme in the other one. And if you pass D-glucose through the middle, the hexokinase obviously will chemotax first because that's its immediate substrate. The second enzyme has to wait until hexokinase makes its substrate, which is glucose 6-phosphate, and then it chemotaxes and so on. And aldolase would be the slowest. And you find that happening. Hexokinase goes in first and then aldolase more slowly. So this is actually an example of nanomotors as information processors, these cascades, because they're coupling information in the form of gradients to direct motion and assembly. And this is, I think, quite significant because the important question is, how does active matter organizes? Obviously, you need a source of information. So the question is, how does the information arise from energy through chemical gradients? And chemotaxis is an example of that. So for example, if you have 1,000 proteins in solution, and if I add a substrate that is add a substrate which is a substrate for the first protein, it will chemotax, and only those proteins which are related in a cascade will come together, and they will come together in a sequential manner. And so you can have directed sequential assembly, and clearly that's an example of information arising from the energy which is coming out from the turnover, catalytic turnover, which is causing this to happen. And one thing I should point out that both the motion and the communication arises from the chemical gradients. The enzymes wouldn't move unless you had these chemical gradients, and neither would they communicate through chemotaxis. And this is something that the physicists need to take note of. Often they use the so-called Vischek model to model movement in active systems. And what they basically do is they assume that things are moving, and then they turn on an interaction parameter. There is no explicit energy input in those systems. And you have to realize that both the motion and the communication comes from the same source, which is the chemical gradient. This is kind of a fun thing. There's a Lambda limit for robotic manipulation. And basically, if you want this red and black balls to react together, you need to recognize, grasp them, bring them together, cause them to react. And according to the Lambda limit, to double the reaction rate, the robotic manipulation requires kBT ln2 energy expenditure. Chemotaxis is basically doing the same thing, right? Because it's bringing two things together through these chemical gradients. And so I thought I'll just throw this out for those of you who are into these aspects of robotics to think about. And the other thing that comes out directly from all this chemotaxis work is that, is that it allows you to direct motion, for example, in drug delivery. And so Daniela Wilson and Samuel Sanchez and many, many other people have worked on systems where you have these particles which can act as containers for drugs. And you can also have enzymes in them, and they can be moved through catalysis, and they will move directionally through chemotaxis. You can shut off the opening so that the substrate can't reach it, and then these things stop. And both positive and negative chemotaxis has been shown in these systems, including the very significant work by Battaglia, who showed that you can use these to cross the blood-brain barrier. Now, this is an area that's very active, and we have written an essay recently talking about some of the issues involved, such as biocompatibility. Obviously, the motors, the chassis, the fuel all have to be biocompatible. They have to move through body fluids where you have blood flow, so they have to be powerful enough to move against blood flow. Maybe they can rheotax, which is movement along the wall where the flow is not that strong because of the no-slip boundary condition. And particles have been shown to rheotax. We have shown some, and others have. They have to move through complex non-Newtonian biofluids. They have to overcome interstitial pressure because cells have interstitial pressure, and to penetrate the cell membrane, you have to overcome that. You have to target precisely. And remember, to go from the circulatory system to the tissue, there may be one kind of fuel in the blood and a different kind of possible fuel in the tissue. So these motors have to be versatile, be able to use different kinds of fuel as they navigate through the system. I like distributed functionality because it's very hard to have a single particle with all the desired functionality. But if you have a population, two populations, for example, one population of particles will search out a target, communicate to the second population, which can then go and deliver a drug or something, that would make life a lot more easier. And then, of course, scale up. So we wrote this essay in small. There have been many other review articles. Wei Wang published one in Advanced Healthcare Material, which I particularly like because he also points out many of the shortcomings of the current approaches. Anyway, I don't want to talk too much about it, except to say that one of the big problems is navigating the circulatory system. But if you don't have any flows, for example, blood flow, then life is a lot easier. So where there's no flow, for example, in GI tract, in the bladder, you can use nanomotors much more efficiently to target disease sites. So for example, Samuel Sanchez has used urease-powered motors for targeting bladder cancer. Joe Wang has used motors in the GI tract for therapeutic purposes and so on. Okay, I want to turn next to the rheology of fluids, changing the rheology of fluids by active particles. This is something I think has not been looked at too much. There is, of course, a lot of information in the literature about how bacteria can change the viscosity of fluids in which they're swimming. Igor Aronson and others have shown, for example, that swimming bacteria can drop the viscosity of the solution by as much as 80%. And more bacteria you have, the greater is this effect. You can also see that bacteria will enhance the diffusion of other things present in solutions. For example, you see a correlation between the active diffusivity of passive diffusivity of tracers versus the active flux from the bacteria, which is basically the number of bacteria times their speed. And then Tom Malouk wrote a beautiful paper during his sabbatical in France, where he showed that even gold platinum nanomotors will also do this, will change the rheology and the diffusion of tracer particles in solution. But the important question is, okay, so these are all micron size objects, the gold platinum rods, the bacteria, and so on. And the question is, can individual catalyst molecules, so now we are down to nanometer size, can they influence the motion of surrounding molecules and particles? We've seen examples of this. For example, we have seen that tracer particles will show active diffusion if you have an active enzyme present. But I think, and so you can couple the motion of catalysts and tracers to allow particle-driven assemblies. But I think perhaps the most interesting thing is this paper, and there are several other papers like this, where people have shown that, in fact, the enzymatic action increases the motion of things within actual living cells. And this paper in particular by Christine Jacobs-Wagner talks about how, and she looked at E. coli cells, that the bacteria cytoplasm, if you turn off all the enzymes, not just the motor proteins, but all the enzymes in an E. coli cell, you can do this by adding dinitrophenol. Then the cell becomes glass-like. So it becomes really stiff and glass-like. And it's only when you turn on the enzyme that the cell becomes fluidized. And the way she showed it is she put tracer particles, in her case, the tracer particles were inactive virus particles. And she showed how they diffuse depending on whether you've turned off all the enzymes in the equalized cell or not. So you can see it here. In the untreated cell where all the enzymes are active, you can see the virus particles diffuse rapidly all along the cell. But if you turn off the enzyme by adding the dinitrophenol, basically they don't. And so this idea of changing viscosity on the fly is an important one that has many possible applications. And this is something that needs to be looked into in more detail. I want to talk now about analyte triggered pumping. And this is trying to couple sensing with microfluidic pumping and to be able to deliver cargo into specific locations. And we've showed this with enzyme pumps. Enzyme pumps, I told you that enzymes show enhanced diffusion and generate force when they're in solution. Now, if you attach the enzyme to a particle, if you attach the enzyme, you can attach it to a particle too, actually. But if you attach the enzyme to a surface, then the enzyme cannot move. And so the force generated gets transferred to the fluid around it. And so the enzymes act as miniature pumps. They move the fluid around. And it's easy to show this. You just attach the enzyme. It doesn't matter how you attach it. And you add tracer particles so you can track the fluid flow. And then you add the substrate and the fluid will flow. And depending on the enzyme, the fluid flows towards the enzyme attached patch or away from it. And if it's a closed system, then you have convective loops. And so we've looked at this, and here are some examples, and you see the video of the catalase pump, for example. The point I make here is just like the enzyme diffusion, the pumping rate varies with the reaction rate. So if you add more substrate, the more is the pumping velocity. Now, these pumps don't last forever, of course, because you're eating up the substrate. But you can recover the pumping velocity if you add fresh substrate. So, for example, in the catalase case, you can see that the rate of pumping goes down. But if you add more hydrogen peroxide, it goes back to the original value. Same with the urease. The rate of pumping goes down. If you add more urea, it goes up again. Okay. So these pumps are rechargeable, if you will. The fuel here is the substrate. And so these are actually sensors come pumps because they only pump when they sense their own biomarker or substrate. So it combines sensing and microfluidic into one self-powered device. And you don't need any outside energy source to pump the fluid. It's self-generated. So you don't need an external power source. It'll turn on whenever it sees the substrate. And so there are some interesting applications of it. For example, you can use it for drug delivery. So for example, one of the things we showed in our Nature Chemistry paper is that you can take a hydrogel, fill it up with insulin, and you can put glucose oxidase, you can attach glucose oxidase to the gel, and you can immerse it into a solution of glucose, which is a substrate for glucose oxidase. And the glucose oxidase pump then gets turned on because of the presence of glucose, and it pumps out insulin. Now remember that the pumping rate depends on the substrate concentration. So more glucose you have, more pumping, more insulin is pumped out. So it's an insulin delivery on demand system, if you will. Some other example, we had funding from our Defense Threat Reduction Agency, which is interested in neutralizing nerve agents. So in that case, nerve agents poison gases are organophosphorus compounds. So the way to neutralize them is to hydrolyze them. And so in this case, our enzyme was phosphatase, which hydrolyzes nerve agents. And so we attach phosphatases to a gel. We put an antidote in the gel. And we used a mimic of nerve agents, glycerophosphate as a substrate. And of course, the enzyme gets turned on because it hydrolyzes the glycerophosphate. So it pulls in the nerve agent, hydrolyzes it, and at the same time, it pushes out the antidote. And you can see as you add more and more of the substrate, you get more and more pumping out of the antidote. You see some background leaching of the substrate, but you can control that by playing around with the hydrogel scaffold, and you can get to pretty low background and still be able to pump out what you need. Interestingly, of course, at Penn State, we use glycerophosphate. It's a nerve agent mimic. It's not a nerve agent. Obviously, we don't use nerve agent at a university. We don't want to kill our graduate students. But the defense agency was interested in that. And so we told them how to make one of these things, and they have all these mutant enzymes that work on nerve agents. And so they made a pump using one of these mutant enzyme, organophosphorus acid anhydrolase. And in this case, these will pump using soman, which is one of the nastiest of the nerve agents. And will neutralize the nerve agents. So you can use these to deliver drugs, antidotes, and at the same time neutralize something that's bad for you. You can also use these as sensors for toxic substances that inhibit the enzyme. So for example, mercuric ion inhibits urease. And so by looking at how the urease pump slows down as you add more and more mercury. Ooh, how did that happen? As you add more and more mercury, you can figure out how much mercury is present. And we can detect mercury to parts per billion level. It's very simple. You can do this on your cell phone camera. You can just look at how the pumping rate is slowing down and figure out how much mercury is present. You can do this for cyanide. Catalase is inhibited by cyanide. And again, we can look at parts per billion levels. So these are levels that are much lower than what the government will permit in your drinking water, for example. You can use other sensors, for example, acetylcholinesterase, which cleaves the neurotransmitter acetylcholine. And this enzyme is inhibited by nerve agents. And so if you use an acetylcholinesterase pump, and again, it's simple, you just have a surface on which you attach acetylcholinesterase and you see the pumping in presence of acetylcholine. And if the enzyme is inhibited, then the pumping is lower. And so it allows you to quantitatively test for the presence of nerve agents and whether antidotes are helping or not. Okay. One other thing I want to point out is that you can also use these pumps to build very sensitive sensors. Now, there is, of course, we live in the world of miniaturization, and so sensors are being miniaturized. You're going from a large sensor to a micro sensor to ultimately to a nano sensor, for example. The problem is that as you make the sensor smaller and smaller and smaller, it gets harder and harder for the analyte that you want to detect to find the sensor. Because the analyte would normally find the sensor by simple diffusion. And if the analyte is somewhat large, it could take days before it can find the sensor that is very, very small. And you can see it here. If you just rely on diffusion, then as I said, it can take hours or days before a large analyte, for example, a virus particle or a spore can find the sensor. If it's a small molecule, it's not such a problem, but if it's anything larger, then it can take very long to find a very small sensor. However, if you put the sensor inside an enzyme pump or any other catalytic pump, then that pump will actively pull in the solution. And this allows for the analyte to find the sensor much faster. So if you have a directed transport, 1 micron per second, for example, you can see that you get much faster detection and much higher sensitivity compared to if you just relied on simple diffusion to find these things, these sensors. Okay, so I'm getting towards the end. I think Wei wanted me to talk for 45 minutes or so, and I do want to leave some time for questions. So I again want to impress upon the idea of systems materials, which is the study of interactions and feedback between individual components of the active system. And the important thing is to try to answer these questions. You know, what kind of systems do we really want? Do we want, for example, modular units that you can take off the shelf, so to speak, and have them self-assemble into active material? Do you need an external programming and control? And as I said, the most important thing is how is the information stored and how is it communicated? Now, the field of nanomotor has grown tremendously since Tom Maluk and I, for example, published the first examples of synthetic nanomotors, experimental systems. People, of course, have been looking at biological motors much longer than that. But to solve some of these problems, you need a unified approach that brings in chemists and physicists and engineers, chemical engineers, biomedical engineers, mathematicians, and everyone together to solve these problems. So in that sense, it's an extremely interdisciplinary subject area. And so I want to end by showing how you can have a way of bringing people together to work on one particular problem involving active systems. And the example I want to show involves catalytic pumps. The enzyme pumps are, for example, and the advantage of catalytic pumps, enzyme pumps, is that you don't need an external power source. You can turn them on and off at will, and you can use this for a variety of applications. And so what I show here is, for example, a multi-investigator center for autonomous fluid devices for distributed manufacturing and diagnostics. So what do I mean by distributed manufacturing? So a lot of the manufacturing is, of course, centralized. Wouldn't it be nice if for example, you had distributed manufacturing where if someone needs a certain chemical, he or she can make it on their own in the neighborhood rather than travel 50 miles to get it or order it through the mail and so on. Same with diagnostics. If you can test for some disease in remote areas. One of the nice things about this autonomous pumps is since there's no external power supply, you can use these in third world countries where power may be hard to get hold of. So this might be easier to do. So in this case, you can see you start with fundamental knowledge, which is a combination of theory, experiment, and design. You look at how catalytic cascades behave to feed each other. You have to know chemical kinetics and flow synthesis, and that naturally leads you to think about what materials and what sensors you want to aim for, what kind of modeling, what kind of interfaces. There are, of course, things that need to be overcome, understanding mass transport, spatiotemporal partitioning, adaptabilities in non-aqueous environment. And once you understand this, then you can start to build the technology base. So you can have control over flow, you can have chip-based autonomous microfluidic devices, you need the software to run them, and telehealth platforms and so on. And here again, you have barriers that you need to overcome, like design tools, flow control, streamline the engineering, and so on. And once you get to that, then you can go the next step to the product distribution diagnostics. And here you have to involve the people who are going to use it. So the individual users or industry, policymakers, health insurers, and so on. And basically, this will give you social benefits of medicine and materials for all. And it's distributed. So the end user is really the producer, or the end user is the one who's using the diagnostic. So you get a feedback loop. What are the opportunities, and what you can feedback, how can you improve the system, and so on. So that's just an example of what can be achieved if people from different fields got together. Clearly, you need chemists to study catalytic cascades. You need materials and engineers for materials and sensors. You need mathematicians and fluid mechanicians for mass transfer problems and so on. So this is the ultimate dream in the area where you have these motors moving through human capillary, fixing problems of various kinds. We are, of course, very, very, very far away from these. So I like to say that we are in this stage. This is what the computer mouse looked in 1968. So that is what, 53 years ago. And the computer mouse has come a long way in 53 years. So similarly, nanomotors, the experimental realization was in the mid-2000, it was 2004. So this field has come 15 years since then. So give us another, I don't know, 30 years, and we'll see what happens. So I will stop here and thank again for listening, and I'll be happy to answer questions.
Okay, thank you, Professor Sen. Thank you for your wonderful and inspiring talk. And now we are going to the Q&A session. And if you have questions, please feel free to unmute yourself and directly talk to Professor Sen.
Okay, so there are a couple of questions in the chat. One question was, when you use catalytic pumps to convectively pull analytes to overcome diffusion, because there's a small window of time for the analyte to react with the sensor. It depends on what pumping system you use. You can have slow, you can have fast, you can have, and so you can fine tune the window by using different catalytic pumps. And I talked about enzyme pumps, but of course, everything I said is general. For catalysts in general, you can use inorganic catalysts to do the same kind of pumping. This has been shown by us, by Henry Hess, by Mari Ashplantu and others. And so you're by no means tied to just enzymes. So the second question was, how long do the enzymes live? You're right, absolutely, that enzymes can die on you. And so obviously over time, they will start to degrade, which is why I say that in some cases, you may not want to use enzymes. You may want to use other kinds of catalysts. There's a lot of work, for example, in China and others, on nanozymes, which are inorganic analogs of enzymes, which are much longer lived. So instead of catalase, which converts hydrogen peroxide to water and oxygen, you can use cerium oxide, you can use platinum, silver, and other ones that are much longer lived. But those are good questions.
Max, do you have a question?
Yes, so sorry, thank you for the talk. I was wondering, so when you listed some of the applications, I noted that you didn't mention, for example, these micro mixing work that was, I think, first done in the Joseph Wang group, and then now a bit more Primero with the sort of environmental pollution degradation, where they say that the motion of these micromotors or nanomotors can enhance localized mixing. Do you see this as a promising field? Like, I'm curious about your opinions on this.
Yes, I think that is correct, and that's an excellent point. I think it's a combination of motion and pumping, which is basically mixing, which speeds up reactions in cells, in living cells that Christine Jacobs-Wagner and others have talked about. Because, as you know, our cells are extremely crowded environments. We have arguably 40% of the fluid in the cell are long polymers, and that slow down diffusion and so on. And it is this turning off of enzymes causing enhanced mixing that allows reactions to proceed much faster.
Thank you. And do you think that this could be applied in other, like, for example, reaction engineering type applications where you have catalysts that might mix for a solution, or is this?
Yes. In fact, we have a paper on review, and the point of the paper is the following. We know that catalytic reactions causes flow. But the flow can, in turn, bring the reactants to the catalyst. So there's actually a feedback loop between the flow and the reaction kinetics that is not appreciated very much. And so one of the things we suggested in the paper is that chemical engineers might want to start thinking about how best to design the reactor systems to maximize the catalytically generated flow. So, for example, if you have catalyst beds in these reactor systems, you could design the architecture of the reactor to maximize the flow.
Thank you very much.
Thank you. One question: what's the potential of chemical motors for bioapps for real clinic use? Like I said, at this point, I think the two biggest problem, and this is just my subjective view, this is not my field, is that it's difficult to go. The motors that we have now are not powerful enough to go against blood flow, And second, if you want to go from blood to tissues, you have to use different kinds of fuel, and most motors use one kind of fuel. They're not motors that can work on many different fuels at the same time. And so the first applications would be where we're not worried about flows, we're not worried about multiple fuel systems. So that's why I brought up the issue of bladder cancer that Samuel Sanchez has been working on, and GI tract that Joe Wong and others have been working on. But not my field, I don't know.
Professor, I got a question from my books. Lei Wang asked, why do you build up the microfluidic systems to check the chemotaxis behavior of enzymes or enzyme nanomotors rather than using static systems? And how will the shear force influence the chemotaxis?
It's a good question. Yes, you're absolutely right. We have not used static systems, but others have. For example, the Battaglia, who published on the chemotaxis of glucose oxidase attached particles towards glucose, he used a static system. What he did was he had a source of glucose at one place in the Petri dish, and it slowly leached out the glucose. And then he showed that the glucose oxidase particles will move towards those. So people have looked at, and Professor He has also shown motion, chemotactic motion in static systems. So others have looked at static systems. And you're right. There are many different ways of looking at chemotaxis. We just happen to have looked at fluidic systems, but we haven't looked at the other ones. And yes, so Wei's question about shear flow is a good one. I don't know the answer to it. Max, you just want to ask?
Sorry, I was just wondering do you have an archive link for this publication that you mentioned, or will be is in review?
Which publication?
You mentioned with regards to the diffusion of these reagents to the surface of the.
No, I don't.
Okay.
I don't.
Is there a title to look out for?
If you shoot me an email, I'll try to send you something.
Okay, thank you very much.
Other questions?
Any more questions?
Yes.
Can you hear me, Professor?
Yes, I can.
Yes, I want to ask a question about the system's materials.
Yeah.
When you want to define this, I wonder if you want to say that you want to build an intellectual nanomotor community that can sense the changes in the environment and react to the environment and even send information to our people.
Yeah, sorry, what is the question? I missed that.
I mean, you want to build an intellectual nanomotor system, right?
Yeah. So
what's the functions?
So what I'm trying to say is that instead of looking at specific components, right? Mm-hmm. You have to have an integrated view of the whole thing. So for example, instead of just looking at motors and how they move, you need to also take into account the fluid through which they're moving. You need to take into account other motors that are maybe present. There may be different kinds of other motors and how they will interact, what kind of chemical gradients will cause them to interact. Can you have feedback loops? For example, so let's say motor A sends a signal to motor B, and motor B in turn sends a different signal to motor A, and then they can communicate. So it's something that is sort of looking at the whole thing instead of individual parts.
Yes,
And
but
so
like
I'm just trying to popularize the term. That's all.
okay, I just think the systems materials is a big work. That is quite hard to
Yeah.
finish right now.
Well, yes, it is hard.
And there are so many functionalities that a macromotor need to equip with.
Yes, but you know, that's something, for example, that systems biology deals with in their system. Their systems can be quite complicated. Now, we don't know enough about the motor system. It's much younger.
Yes, exactly.
Yeah. There was a question about chemical oscillations from Jinyao. Jinyao, it's nice to hear from you. And yeah, and I didn't touch on chemical oscillations at all. And it is a very interesting thought. And let me point out something in that regard. The chemotaxis is kind of a continuous system. There is no threshold. So motion in response to a chemical gradient of the type I talked about, there's no threshold. But if you had an on-off threshold, then you can do a lot more interesting things. So these would be like switches. They'll be on or off. You can have quorum sensing and other ways of doing things. Oscillations of different kinds, pattern formation, which might lead to new interesting metamaterials that have interesting optical properties and other properties. Yeah, there are all these areas that I don't know enough about, and so on.
Okay, thank you.
Of course, Samuel had to ask something. Useful to ask, because a different part, different. Yeah, ideally, yeah, if all the particles have the same type, they gave out the same chemical gradients. It's interesting but boring. That's not what living systems do. And so there has to be give and take. If you'll indulge me, there is this thing called Turing test. And the Turing test is that you are sitting and across from you, there is something, it's a human being or a computer, and you can't see it. And all you can do is you can ask questions and get answers. And from those answers, can you tell whether the person you're addressing is a human being or a computer? If you cannot tell the difference, then that system has passed the Turing test. There's a bacterial version of this where you can have for example, a protocell, a synthetic cell, and a bacteria. And the bacteria sends a signal, the protocell processes it, sends a signal back to the bacteria, and the bacteria then looks at it and says, hey, that's another bacteria, doesn't know that it's a synthetic cell. And some of the work in this area, beautiful work, has been done by Mansi and others. And so that's an example of different populations interacting through this feedback, give and take loops. One of the things, many of these things would require is a memory. And And I have no idea how to build memory into these systems, because memory requires not just temporal, but spatial abilities. Okay, that's a long answer to your question, Samuel. Did I say swarms in the bladder are boring? You should be working on the swarms instead of asking me questions. Okay, there's one more message. No, I don't know. Any other questions?
Is there any more question? Someone raise their hand.
Yeah, Max, ask. Guess not. Okay.
Professor, there is another question in the chat box.
Oh, there's another question in the chat bar. What is the difference between an external device and a motor? The difference is that the motor harvests energy locally. You're not tied to a source of power. This is actually an interesting point because, most sensors, or not most, all sensors now are tied to a power source, and therefore they have limited area that they can sample because they can't go very far, they can't detach themselves from the power source, etc. And in this case, the motors being self-propelled and harvesting energy from the surrounding can do certain things that you couldn't do using external devices that need a power source. So that's what I think is one of the advantages.
Any more question?
And by the way, I want to thank Wei and his group for hosting the Nanomotor update talks, but also all the updates. I find many of the papers through these Nanomotor updates tweets. And so I need to thank you all for doing the service to the community. Oh, one more here. Too bad that you have to buy another 35 years to buy. Wei, you are one of the ones who wrote this pessimistic review article. That's okay. But believe me that people are looking at this. I've had people from companies, people from the US defense agencies talk about using these in the field and so on. Another application they talk about is smart dust, which are these sensors that you can have small particles, microparticles, that you can spray from an aircraft onto fields, and they can detect, for example, vapors from explosives, other things, and they will be all self-powered because there's no power source when you in these remote areas. So that's just another example. So there are many examples that you can think of. You just have to be creative and have to work with people in other areas who know things that you don't know and I don't know. If you had $1 million if you had $1 million to invest, which application would you bet on? I will tell you if you give me $1 million.
Any more questions? If no more questions, we would like to thank Professor Sen again for his wonderful and inspiring talk. And
Thank you so much.
Yes,
Take care.
Thank you. And thank you all the audience for listening.
Thank you.
Hope see you next week.
Okay.
Okay, bye-bye.
Thank you, Ayusman. See you.
Thank you.